The overall goal of the eye irritation test is to classify and label substances and mixtures according to their eye irritation potential. This procedure utilizes a three-dimensional reconstructed, cornea-like tissue model. Upon arrival, the test kit is pre-incubated overnight in the assay medium. The following day, the tissues are hydrated, followed by the topical application of the positive and negative controls and the test chemicals of interest onto the surfaces of the tissues in duplicate. After a fixed period of exposure, test chemicals are removed by washing. The tissues are post-incubated in fresh assay medium, and the toxicity of the chemicals is then evaluated by MTT assay. Ultimately, the irritating potential of the test articles can be assessed by comparing the relative viability of the test article-treated tissues to the negative control-treated tissues.
The main advantage of this technique over existing methods, like rapid trace irritation test, are that it does not involve laboratory animals. It utilizes in vitro reconstructed cornea-like tissue model epiocular, which comprises normal human cells. It's applicable to all types of substances and mixtures and can discriminate between chemicals that induce ocular irritation or serious eye damage from materials, non-irritants, that do not require classification and labeling.
The reconstructed human cornea-like epithelial tissue, or epiocular, is prepared in inserts with a porous membrane through which the nutrients pass to the cells. Reconstructed tissue forms a non-keratinized epithelium.